magnetic brain stimulation for MS, the TAURUS journey. This is really a fascinating tale that started in the lab and has ended up in the clinic, which is what we call translational research. We're translating the findings that you've seen all talk about cells and stem cells and whatnot. What is it? How can we actually apply those findings into humans to try and improve MS? And this is an example of how we can do this. This is how we've taken something that was discovered in the laboratory and taken it into the clinic. So, people living with MS have identified myelin repair as one of the biggest unmet needs. If we can actually correct the damage before it becomes irreversible, you can potentially improve people's quality of life and also reduce their disability. But that's an unmet need. Currently, there's no treatment that's been shown to actually improve or repair myelin. Yeah. The ultimate goal is clearly to develop treatments that induce myelin repair. And one promising approach is to use magnetic brain stimulation, or MBS. And in this graphic, this is just to show, and some of this has been shown before, it just shows that how a neuron, which is the cell that's at the end there, which glows, and then you can see what's called the action potential travelling down the axon, which is the blue and yellow thing there. And as demyelination occurs, and we don't know exactly the mechanism of how demyelination occurs, but it's thought to be antibody mediated or a direct damage from the cells, the pink cells surrounding. You can see that as you demyelinate, the action potential, which is is the information package, if you like, that travels down the neuron, slows down and eventually that you don't get any transmission. And when you don't get any transmission, often you'll lead to cell death, and that is irreversible. Once you start losing neurons, you never get them back. So if we could actually induce this cell to remyelinate before it undergoes irreversible cell death, that would be a really major advance. Now, why magnetic brain stimulation? Why should this technique improve myelin? It's non-invasive, and it's a way to change nerve excitability. So when you give people magnetic brain stimulation, you're actually stimulating all the nerve cells within the region of where you're giving the stimulation. And we do this by using a coil, as you can see here demonstrated, over the vertex of the head. We can do that on both sides. We can do it on any part of the brain, and this coil can deliver stimulation at varying levels, and sometimes to the point of being intolerably high. But what we tend to use clinically is stimulation which is not really detectable by the person. It works by, if you all remember your physics, that when you generate a magnetic field, you get a current generated at the right angles to the field. If you all remember your grade 9 physics, is your field, force and current. It works this way. So we can induce a current in the brain, which stimulates the brain cells, and we can do this by putting a coil or a magnetic, really quite powerful magnet on the surface of the brain, and then we can use different stimulation pathways, but the one that Kayleen's group found to be the most used was theta burst stimulation. And this induces, as you can see in the thing, each cell to fire off, and then it will transmit between cells, and it can stimulate the whole brain. Even though when you're having it, you don't necessarily know that your brain is being stimulated. And this technique has been used to study a whole range of things in medicine, within the human system, and also within the and to be therapeutic in things. Different ways of doing MBS has been found to be therapeutic for things such as depression and also obsessive compulsive disorder, pain disorders. So it's a very, very useful technique. So, and why how does MBS promote remyelination? And what we do know from our laboratory studies is that we do have immature or oligodendrocyte progenitor cells, which are the red cells, which are not myelinating. And if we could stimulate those cells to become myelinating cells, which is shown here by the white cells, which you've seen many pictures of oligodendrocytes, you see them ensheathing the axon in this cell. And if we can actually show that this is now demyelinating that axon, and if we could then induce these red cells to suddenly become myelin-presenting cells, myelin-producing cells. That is a way of actually inducing remyelination. And we know from the laboratory work that Kaylee and her colleagues have done is that this is one of the mechanisms by which MBS can stimulate remyelination. And I'm just trying to get this to move along, and it's not doing it. All right, so here we go, and we're going to see the cells, the red cells, we can show increase in number, and then the red cells then become myelinating cells, and it allows the, here we go, now we're going to remyelinate, and suddenly we can restore action potentials to that cell. And so this allows, so by this mechanism, MBS we can show that in the animal model, that we can induce remyelination in an animal model of MS, which you've seen described, the Kiprasone model and other models, and that this is potentially a way of being able to repair myelin in the human brain. And we can see here that this is from the lab to the clinic. We can see that this is a mouse on the far left, having MBS. Kayleen's been able to publish quite a number of papers on this subject, which are really groundbreaking. And then that's then been translated into the clinic, which is where I become involved. And we've done 2 TAURUS trials now. TAURUS 1, which is a phase 1 trial, and you always hear about phase 1, phase 2, and phase 3 trials to see whether something works. And a phase 1 trial is basically not to show whether your treatment works. It is to show whether it's safe, and the protocol that you're going to use is acceptable to people with the condition, and that it is feasible, and that you can blind people so they don't know whether they're having MBS or they're having a sham treatment, because that's the way we determine whether something really works, is by being able to do a clinical trial. So we've done TAURUS 1, which is a phase 1 trial examining the safety of magnetic brain stimulation for people living with multiple sclerosis, because no one had ever done this before, that we did not know what would happen if we did this theta burst treatment on people with MS. What would be the short and potentially long-term effects of it? It's, it comes up with this fancy acronym, which is magnetic. That's how we got TAURUS out of magnetic brain stimulation for multiple sclerosis, the TAURUS 1 trial. We've registered this with all the appropriate government agencies, and the trial registration with ANZ Clinical Trials Clinical Trials Registry, and we designed this as a randomised controlled trial. It was double-blind, sham, placebo-controlled, parallel group safety trial, and we wanted to know, as I said, if MBS is safe and tolerable for people with MS, to determine if our protocol was feasible. Would people come in and have this treatment? To obtain preliminary data about the effects of MBS on MS symptom severity, fatigue, quality of life, anxiety, depression, motor and cognitive function, and to obtain preliminary data about the effects of MBS on myelin repair using advanced MRI techniques. And that's one of the important parts, is that we had to actually develop an MRI technique which measured myelin density. And we sought a population of people, and inclusion criteria. Obviously, to go in the trial, you had to be diagnosed with MS and had to be relapse-free for 3 months. You had to be between age 18 to 65. Your measurements of disability had to be between 1.5 and 6. You were willing to be able to come in each weekday for 4 weeks, to have 20 treatments, which was shown to be the effective amount in the mouse model. You had to be capacity to provide informed consent, and we had a number of exclusion criteria. We did not want to give this to women who are pregnant or intending to become pregnant. We wanted to make sure that we weren't going to cause any unexpected adverse events in people who had other brain conditions. We wanted to make sure that the people who were in the trial were able to have an MRI scan, because this was a critical outcome measure for us. And we didn't want them to know what MBS was, so we didn't want them to have had magnetic brain stimulation for other conditions. And we wanted they couldn't be currently involved in another trial. They had to have access, for this first trial, to access to a smartphone or the internet to provide some of the outcome measures. And they had to be able to understand the trial in English. So the trial design, we recruited 20 participants in Hobart. 2/3 of them got MBS and 1/3 got placebo. We did an MRI pre-treatment and all of our surveys. They had MBS or placebo treatment, 20 daily sessions, Monday to Friday, 15 minutes per day. And post-treatment, they had an MRI and surveys again. So what did we find? We had 5 adverse events and 1 serious adverse event, which was someone who had to be admitted to hospital. None were related to the MBS treatment. Protocol compliance was very high. We had 85% of people complete the protocol as defined. Blinding was successful. People could not tell if they were getting MBS or placebo, and they really struggled hard to figure it out. They really wanted to know whether they were getting the active or the non-active treatment, but they could not tell. MRI measures and MRI symptoms did not change over the 3 to 4 month trial period. So we found the conclusions in this study were that 20 sessions of low-intensity MBS using an intermittent theta burst pattern is safe and well tolerated in a small number of people with MS. The study protocol and procedures are feasible. We needed to improve our placebo because what we found was that even though the manufacturer said there was no stimulation if we held the coil at 90 degrees or flat, it was in fact getting some stimulation to the brain. So we weren't in fact giving a true placebo. So we had to really look at that. We also found that the Floodlight app, which was the smartphone app that we wanted to use, was not a suitable measure of motor or cognitive function in MS. We had multiple issues with the Floodlight app, and therefore we decided that we would not continue with it for the phase 2 trial. This was recently published in our results. And then we moved to TAURUS 2, which is a phase 2 trial examining the safety and preliminary efficacy. And this is where you start looking for efficacy. You want to know whether your treatment works. And then you've got to do a larger study using a larger number of people. Again, it was the same acronym for TAURUS, but this was just with a 2 on the end. And we registered it, we ran in trials, and it was a randomized sham, placebo-controlled, blinded, parallel group safety and preliminary efficacy trial. So what did we do between TAURUS 1 and TAURUS 2? We changed the sham protocol, so instead of in TAURUS 1, we put the coil in for the active position, and the sham position we turned it to 90 degrees. With this one, we basically put the coil on the head and we put it on the treatment intensity. And for those in the sham group, we put it down to 0. But we played the same noises to the person, so they were unaware of whether they were getting active or inactive treatment. But it meant the operator who gave the trial, as in the first trial, knew whether the person was getting sham or active treatment, and they were not involved in any way with the future analysis. We changed the Floodlight app to the Multiple Sclerosis Functional Composite Score, which included a 25-foot timed walk, a 9-hole peg test, and single-digit modality test, which many of you will be familiar with who've been involved in our trials before, which are standard measures of MS. We added measurements related to sleep quality to our secondary outcomes. Recruitment was at 6 sites around Australia because we couldn't do this all in one site. We had the Menzies, we had the Launceston General Hospital, the Alfred Hospital in Melbourne, John Hunter Hospital in Newcastle, and the Mater Research in Brisbane, and the Perron Institute in Perth. And so with 6 sites, we were able to recruit a significantly larger sample size. We used the same inclusion-exclusion criteria in TAURUS 1 with very minor tweaks. We trained all of the sites face-to-face in how to do the protocol, and that was really important to maintain consistency across the sites. MRIs were standardised, again using the Sydney Neuroimaging Assessment Centre. Statistical analysis of what we call power calculations indicated that we needed to recruit 108 people, allowing for a 20% dropout rate to get a number of people to measure a 20% difference between active and placebo. We anticipated a relatively high dropout rate because we were still, when we were designing this, we were still in the throes of the COVID epidemic. The 108 participants gave us ability to measure a 20% difference in the MSFC between the MBS and sham patients, again using 2/3 in the active group, 1/3 in the placebo group, with 80% power, which is a perfectly adequate amount for a trial of this type. So 20 sessions were given as per TAURUS 1, and our hypotheses were that 20 sessions given over a 4 to 5 week period delivers an intermittent theta burst pattern will improve functional outcomes and symptoms of MS, and so result in detectable changes on MRI metrics that are consistent with increased myelin in the brains of people with MS. So the primary objective is to determine if the MBS improves the MS functional composite score. The secondary objectives were, quite a few of them, was safe. Does it improve quality of life, reduce anxiety and depression, reduce fatigue, improve sleep quality? And our tertiary objectives were that MBS effectively promotes myelin addition to the brain of people with MS. Myelin addition, that myelin addition correlated positively with the functional and patient-reported outcomes. Protocol compliance and adherence to treatment schedule is equivalent for MS, for people with MS in the MBS and the sham, and a sham placebo protocol ensures that participation is blind to, again, we wanted to know that we could actually do this in a blinded manner. So what have we done? So we got lead ethics approval from the Hunter New England Health on July 2022. Our first participant randomised in November 2022, and our last participant randomised on the 13th of May, which is not quite 2 months ago. This study has now closed. All participants have completed MBS or sham treatment and primary, tertiary outcome measurements, which is really important to know. We've finished this trial by the 30th of June, which is our main aim. We enrolled 104 participants. We originally aimed to reach 108, but only 7% of our participants withdrew, which is significantly less than the 20%. So we actually ended up with more people to analyse than we need to analyse than we needed to meet our power requirements. Compliance has been high, 85 to 95%, and data is currently being cleaned, and analysis is now ongoing, and we expect results are expected by the end of the year. Unblinding will occur after analysis, so we'll be able to tell people what they got, and consumer involvement has been in this trial right from the start. We have had, in all aspects, from the design of this trial to the execution of the trial, we've had consumer involvement. CCI has improved all aspects of the design. They've been involved with the community, the patient information and consent forms, promotional videos. Everything that's patient or consumer facing has been developed in conjunction with the consumer committee. All our advertising materials, all our outcome measures as well, study design, feasibility and management. And a special thanks really needs to go to Meg Denham again. She gets a lot of publicity, but she's done an absolutely fantastic job in being involved in this. And it's really, her work on this trial has been outstanding. So what I'd like to do is thank the people, I don't know why I need to thank myself, but all the people who've contributed to the project. In particular, I just really would like to emphasise the fact that this is translational. The Kayleen and Carly and Jenny Rogers from Perth were the people who showed that you could do MBS in animal models. They built the coils. They did all this basic sciences work, and this is a true translational program. I'd like to really thank all the other people involved, particularly people such as Armin and Tram, Kalina, who've all been directly involved with the actual implementation of the trial. Monica Breslin, who's the statistician, Mark Hinder, who's an MBS expert, and Michael Barnett, who's from the SNAC, the Sydney Neuroimaging Assessment Centre. I would particularly like to thank all the participants quite a few people in this room, and notice we've put coils on their head. We would really like to thank the project funding. This is not a cheap trial to do. We've had, the MRFF has provided funding for this trial. We've had the Royal Hobart Hospital Research Foundation, NH&MRC, MS Australia, and in particular, and also the Irene Phelps Charitable Trust and the Medical Practitioner Society of Tasmania. And hopefully next year, when and if we have this meeting, we'll be able to tell you the results of the trial. But I don't know them myself, so I'm not holding back on you. We haven't done the analysis yet. We've just finished this trial. So thank you very much.